Assalamualaikum warahmatullahi wabarakatuh and very good day. Today we will proceed our class science subject to subtopic 4.7, that is plant reproduction. So in this subtopic we will learn about uh, communicate about the structure and function of each part of a flower, justify the pollination process, describe the process of fertilization, and explain the formation of seed and fruits in plants, describe the germination process of a seed, and also solve problems if germination does not occur. So in this subtopic, we will learn about plant reproduction, same as what happened to the animal reproduction and human reproduction. So as you know that every living organism, they have to reproduce to make sure that their generation can be survived, and if not, the their species will die and no more in this world. So what they have to do is to reproduce. So right now we have to look how plant reproduce to make sure to ensure their survival. So first of all, what you have to know that the main organ in plant that take part in the process of reproduction is fruit. So fruit, or that exactly have in the what we call it as a flower, play an important role to make sure to reproduce a new organism. So now flower itself will becoming a part of reproductive system. So the first thing that we will learn is about the structure and the function of each part of a flower. So you have to remember that flower is the organ or reproduction organ that play an important role in reproduction in plant. So now we go to the structure and function of each part of a flower. So as you know, this is the flower. For example, taken from the Hibiscus rosa sinensis, and so the part or the structure of this flower consists of, the first one is this one, is a petal, usually colorful to attract insect and animals. Next is sepal, usually green. This is sepal, usually green in color, and its function is to protect the flower during the bud stage. Next is, okay, so inside at the center of the flower consists of ovary, and inside the ovary consists of ovule. So on top parts of the ovary is this one, we call it as a style, and at the top part consists of stigma and stamen. Okay, right now stamen, this one, the circle one, is a male reproductive organ that consists of anther and filament. So stamen consists of anther and filament. So stamen is a male reproductive organ. And pistil is a female reproductive organ. And this pistil consists of stigma, style, ovary, and ovule. So four components that made up pistil, that is female reproductive organ. So you have to remember, okay, exactly stigma is here, not here, at the center parts, at the top parts of the style. So it's the label supposed to be stigma is here, because this one is exactly the bottom part of stigma. At the side part is exactly a stamen. So don't be confused about this. So stigma is here. Please make a correction. Okay, so stigma here, and then the other parts that look like same as stigma is stamen. So this one is stamen and this one is stigma. So this is the structure of flower, and you have to remember the function that we will discuss after this. So what is the function of this flower is given here also. So I want you to remember the structure and function of each part of flower. Okay, next is we go to the process what we call as a pollination. Okay, what is the meaning of pollination? Okay, pollination is a process of of what is the text? Okay. Okay, this one. Pollination is the process of transferring matured pollen grain from anther to stigma. From anther to stigma. So, when a pollen grain, a matured pollen grain, is transferred from anther to stigma, so we call this as a pollination process. So, if we can look back into the structure of flower. So anther is one of the part of stamen, and this is the position of anther at the top of the stamen that support by filament. So inside this anther consists of what we call as a pollen grain. Inside pollen grain consists of male gamete. So you have to remember, stamen is a male reproductive organ that consists anther and supported by filament. Inside anther consists of pollen grain, and inside the pollen grain consists of male gamete. So what will happen during pollination is pollen grain in this anther will be transferred to the stigma. So this process is called pollination. Okay, so the pollination is a process in which pollen grain inside the anther is transferred into the surface of stigma. So I hope that you understand the main point here about the pollination process. So based on this pollination process, there are two types of pollination that are self-pollination and cross-pollination. Okay, before that, we will discuss about this one. Okay, about type of flowers. Okay, there are two types of flowers, bisexual and unisexual flowers. Bisexual, bi means two. Two sexual means that in one flower consists of two, both male and female reproductive organ. While uni means one, means that that flower only consists of one reproductive organ, whether male or female. So from the diagram given here, we can understand what is the meaning of unisexual flowers. Means that unisexual, uni means one. So one flower is only have one type of reproductive organ. For example, this one. So one flower, this one is a male flower, while another one is female flower. So if that plant consists of flower that is unisexual flower, so they will have a criteria like this. Means that one flower consists of one reproductive organ. So this is the example given. And bisexual means that one flower consists of both male and female reproductive organ. Same as what we have discussed and the example given before. Okay. Example of unisexual flowers. Example unisexual flowers are corn flowers, papaya flowers. Okay, so you have to remember this one. Corn flower and papaya flower is example of unisexual flowers. Okay, so this is the picture to show papaya plant. Male flower is like this and female flower is like this. Same as corn flower also, unisexual flower. Male flower, the structure is like this and the female flower is like this. So we go back to the type of pollination. So as you know that pollination is a transfer of pollen grain from anther into the surface of stigma. So pollination divided into two, self-pollination and cross-pollination. So what is the meaning of self-pollination? Self-pollination means in one plant, the pollen grain is transferred into the stigma of flower in the same plant, whether it is in the same flower or to another flower but in the same plant. So for example, this one, pollen grain from flower A transferred to the stigma of flower B but in the same plant, or pollen grain from the flower C transferred to the stigma of flower C, same flower, so but still in the same plant. So if this happen, so the pollination is called as a self-pollination. Cross-pollination is a transfer of pollen grain from one flower of the plant A to the stigma of flower of plant B. Means that the process occur in a different plant. So plant A and plant B. So if this pollination occur, we call it as a cross-pollination. Okay. Next, okay, I want all of you to do this activity as your homework, activity 4.8. So you have to read the instruction given here, find the information from the internet, and do it in your notebook. Okay. Next, we go into the pollinating agent. In order for the pollination to occur, so exactly flower or the plant need help from other agent. We call it as a pollinating agent. So how do this pollen grain from a plant get transferred from the anther to the stigma of the flower? So we have several pollinating agents that help the process of pollination. So if we look here from the diagram given here, okay, this is a flower A and this is the flower B. So we try to explain the process of pollination that assisted by pollinating agent. So first of all, anther. So this is anther. Okay. So pollen grain from anther will be transferred to stigma of flower B. So how this pollen grain is transferred? It will be assisted by pollinating agent. For example, in this one, the pollen grain is transferred through the butterfly insect. So pollen grain from the anther of flower A stick on the body or leg of the insect. Okay, so when this happen, the butterfly will fly and then land on the stigma. So when this process happen, so the pollen grain from flower A transferred to the flower B on the surface of stigma. So the pollen grain carried by insect are transferred to the stigma of flower B. So this is how pollinating agent help the process of pollination. So what is the other pollinating agent that can also assist in the process of pollination? So for example, wind, animals, and insect are pollinating agent that help in the transferring process. Okay, now we look what are the characteristics of the flower that determine its pollinating agent. So characteristic of flower, exactly what you have to know that every flower have their own characteristic that attract or that suitable with the pollinating agent. So for example, if that flower is the pollinating agent is animal or insect, so it will suit to the animal and insect for the easiest of the to make for the animal and insect to do the process of pollination. For example, if that flower is the pollinating agent is animals and insect, so the characteristic of that flower must be suitable with the animal and insect. For example, the characteristic of animal pollinated and insect pollinated flowers is have big and colorful petals, have nectar and smell nice, and also produce rough and sticky pollen grain. So pollen grain usually stick on the beak or body of the animal. When an insect lands on a flower to suck its nectar, pollen grain will stick to its furry feet and body. So example of animal pollinated flower and insect pollinated flowers are durian, rambutan, papaya, hibiscus, sunflower, and rose. So you have to remember the example of animal pollinated flowers and insect pollinated flowers. Next, second pollinating agent is wind. If the pollinating agent is wind, so the characteristic of that wind pollinated flowers as given here have white or pale petals, have long, furry stigma, plenty of small, smooth and light pollen grain, have a long filament and style. Example of wind pollinated flowers are corn, grass and paddy. So this is the explanation about the pollinating agent. Okay, now we look into a discussion about the advantages of cross-pollination. Between the self-pollination and cross-pollination, cross-pollination has more advantages compared to the self-pollination. Why? Because cross-pollination combines genetic material from two parent plants of the same species. So because of this, cross-pollination has many advantages compared to the self-pollination. So because genetic material, when they combine from different parent plant, so different parent plants will have a varieties of characteristic. So when they combine together, so it will increase the quality, it will increase uh, the genetic of that material of that plant that will be produced. So here is the advantages of cross pollination. You can read from here. Healthier plants, we can adapt better to changes in environment, new varieties of plant, good quality seeds, new plants that are more resistant to pests and diseases. Yeah. So Uh, cross-pollination ensures cross-product inherit good characteristic from both parents. Okay, so that is the cross-pollination advantages. And what the innovation of cross-pollination in agriculture? So in Malaysia, this innovation from the process of cross-pollination is not something new, and mostly the research on this one is conducted by several agencies, for example, MARDI and MPOB. So among the examples of innovation of cross-pollination in agriculture are summarized here. So example given here, plant, palm oil, papaya, corn, paddy, and also other plant that undergo the innovation of cross-pollination in agriculture. So we take only one and the others you have to read it, and please to remember all the name of plant, the type of parent plant, and the plant that is produced, we call it as a hybrid plant. Hybrid plant means the plant that is produced from the process of combination of two parent plant. For example, this one, palm oil. So we take different species, for example, Piscifera, one type of species, and another species dura. So different species of palm oil. So when we combine these together for the cross pollination, so it will produce hybrid plant. We call it as a tinera. So this tinera will have a very good characteristic. For example, more fruit and flesh and have a thinner shell. So same thing happen to the other type of plant, have also the innovation of the cross pollination. So I hope that you can remember all these things. The name of the hybrid plant, Exotica Masmadu Malinja. Okay, now we go to the fertilization process. Okay, after we have learned about the pollination process, that is the transferring of pollen grain from anther into the stigma of the female reproductive organ. So now we will proceed to the what we call as a fertilization. So you have learned about the fertilization process in the human being or in animal. That is the process of combination or fusion of the male gamete and female gamete. So fertilization in plant also have a same concept, that is the combination of male gamete and female gamete also happen in plant. So now when pollination occur, next step is a fertilization. So from here you can see this diagram. Okay, explain a little bit about the fertilization process. So when pollination occur, so from here pollen grain will fall onto the surface of stigma. So from pollen grain consists of a male gamete. So what will happen is pollen tube will be produced, and this pollen tube will growth and bring along male gamete towards the ovule. So what will happen? Pollen group will growth, so and bring along pollen. Uh, sorry. Okay, this pollen grain will produce pollen tube, and this pollen tube will bring along male gamete. So this pollen tube will move towards the ovule and then enter into the ovule. And what will happen? Male gamete inside the pollen tube will enter into the ovule and then will fuse with the female gamete in the ovule. So when male gamete meet with the or combine with the female gamete in ovule, so this is what we call as a fertilization process. So you can see from this diagram how fertilization is occur in plant. Okay, after fertilization occur, so what you have to know is when fertilization occur, so it will produce a zygote. Example that we have learned in human. So this zygote in human will develop to become embryo. So mostly same concept happen to the process in reproduction of plant. So when fertilization occur, so this ovary inside that consists of the ovule that have been fertilized, it will develop to become a seed. Okay, will develop to become seed. So inside a seed consists of embryo wrapped in a seed coat, or we call it as a testa. And then the other parts of the flower will wither and fall. So this seed will develop after the fertilization process, And the structure of seed and their function is given here. So exactly we have two types of seed. We call it as a monocotyledon, monocotyledon, monocotyledonous, monocotyledon, and the second type is dicotyledon, dicotyledonous. So what is the difference between mono and di? Okay, the same word is cotyledon. Cotyledon. The different word is mono or di. So cotyledon here. Okay, before that we have to look into the structure of seed. Okay, cotyledon is the part in which provide food for the development of seed. So one seed maybe have two cotyledon, so we call it as a dicotyledon, and one seed maybe have one cotyledon, so we call it as a mono. So mono refer to one cotyledon. Okay, so the structure of dicotyledons, okay, like this, front view, consists of testa. This is the seed coat. Okay, this is micropyle, a tiny hole, and this is hilum. Okay, longitudinal section, cross section here. Okay, this is testa. Okay, we have plumule here, and also radicle. Okay, radicle at the bottom part, plumule at the upper part. So this is the main structure in the seed. Okay, this is a cotyledon for the food supply, for the development or the growth of embryo. So we call this also as embryo, same as what we discussed in human reproduction system. So this is the monocotyledon. The structure is like this. Okay, inside the monocotyledon we have radicle here. We have plumule, cotyledon, endosperm, seed coat. So this is the structure of monocotyledonous seed. Okay, so for every structure, the function is given here. So testa protects the seed. Hilum, place where the seed stick to the fruit. Micropyle, small hole to allow air and water to enter the seed. Plumule, part of the embryo which develop into a new shoot. Radicle, part of the embryo which develop into the root. And cotyledon or endosperm store and provide food for the seed. So the most important part here is plumule in embryo, that when they develop, it will become shoot. And then is second is radicle, that will become root. So this is the two structure in embryo that very important to know, because these two will become a new plant. Okay, so this is the structure and function of seed. So now we look into how seed become a plant, a new plant. So the process of seed become a plant, we refer to the process of what we call as a germination process. So germination of seed. How germination of seed occur? So germination of seed depend on several factors. Okay, that's the factor how seed will germinate. So before that, we will look into what happened during the germination. Okay, we have two types of germination. The first one, we call it as a apigeal germination. This one, apigeal germination. And the second one is hypogel germination. Eh? Two type of germination process. One is epigel and one is hypogel. Okay, what is the meaning of epigel germination? Okay, epigel germination is a process in which the cotyledon that is being planted into the soil during the process, the growth process, it will come out from the soil surface, same as given in this diagram. So this is the diagram to show the process of epigel germination. So you can see from here, seed is planted into the soil, and when the process of germination, the cotyledon itself will come out from the soil like this, and then new plant is produced. So this is epigel germination. So you can see leaf here and stem from plumule, and this is cotyledon that becoming small because the properties of the inside content of cotyledon is being used for the process of germination. So next we look into the hypogel germination. So hypogel germination refer to the seed with cotyledon that planted into the soil still inside still inside the soil, and the plant will come out from the soil without bring along the cotyledon. Means that the cotyledon still remain in the soil like the diagram shown here. So you can see the process of germination. Plumule, radicle will grow to become root, and then the plumule will grow, develop to become shoot. So now this is the new plant produced from the germination process. Okay, what is the condition required for the germination of seed? Simple, the three factors here. Seed germination required water, air, and suitable temperature. I repeat, seed germination required three factors. One is water, air, and suitable temperature. So this is the most important thing. The three factors must exist together in order for the seed to germinate. Okay, so exactly we have one experiment to study about the factors that affect the germination process. So if we look into this experiment, so the manipulated variable, responding variable, constant variable given here. Okay, so the problem statement are air, water, and suitable temperature necessary for seed to germinate. So the hypothesis for this experiment, seed needs air, water, and suitable temperature to germinate. So what is the procedure? You can see from here. Prepare 4 boiling tubes label A, B, C, and D as shown in figure 4.37. A, B, C, and D. Okay, in tube, boiling tube A consists of wet cotton wool, seed, and covered by sugar paper. Boiling tube B consists of dry cotton wool, seed, and also sugar paper. Boiling tube C consists of seed, cool boiled water, cooking oil, covered by sugar paper. Okay, same thing for D, covered by sugar paper, but consists of wet cotton wool that is put into a different temperature. So test tube A, B, and C are put in lab, while test tube D is put inside a refrigerator. So this seed, we have to wait about 5 days, and after 5 days, we have to take the observation what exactly happened to the 4 boiling tube that consist the seed. So which boiling tube that consists seed that will germinate depend on the factors that we have discussed before. So do you think which test tube that will have seed that will germinate? So you have to discuss this one. Okay. So that's all for the 4.7 subtopic in the reproduction of plant. So I hope that you can do all the notes in this subtopic, and after completed after completing the note in your notes book, so as usual you have to do the formative practice 4.7, answer all the question in your notes book. Okay, after that please read the concept map here in your textbook and also self-reflection. So this one, the last subtopic in chapter 4, we finished already. So means that you have to make a revision for this chapter 4 from 4.1 until 4.7. Please read this summary. And then after that, we have summative practice. I hope, okay, this is the homework for you to do. Please do all the question from question number 1 until question number 9. Sorry, number 7. Okay, so this is the last question. Question number 1 to question number 7. Please do and answer this question in your notes book. Okay, I hope that you can make a revision. Please read all the concept, all the text given in your textbook in order to understand and remember what we have learned previously in the all subtopic in chapter 4. So that's all for today. Happy learning. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.